Hello class, today we're going to learn about the antimicrobial substances. Body produces certain antimicrobial substances, a final component of the second line of defense, in addition to the chemical factors mentioned earlier. Among the most important of these are the proteins of the complement system, interferons, iron-binding proteins, and antimicrobial peptides. The complement system consists of over 30 proteins produced by the liver that circulate in blood, serum, and within tissues throughout the body. Complement proteins act in a cascade, where one reaction triggers another, which in turn triggers another. More product is formed with each succeeding reaction in the cascade, amplifying the effects. The cascade of complement proteins that occurs during an infection is called complement activation. The complement system has three functions. The first enhances inflammatory response, such as attracts phagocytes. The second is to increase phagocytosis through opsonization or immune adherence. The third is to create membrane attack complexes, MAC. As mentioned previously, the classical alternative and the lectin pathways result in complement cascades that activate C3. Activation of C3 in turn can lead to cytolysis, opsonization, and inflammation. Opsonization or immune adherence promotes attachment of a phagocyte to a microbe. This enhances phagocytosis. The classical pathway was the first discovered. It is inhibited when antibodies bind to antigens. Antibody attach to antigens forming antigen-antibody complexes. The antigen-antibody complexes bind to and activate C1. Next, activated C1 activates C2 and C4 by splitting each of them. Then C2 split into fragments C2A and C2B, and C4 is split into C4A and C4B. C2A and C4B combine and together activate C3 by splitting it into C3A and C3B fragments. C3A participates in inflammation and C3B functions in cytolysis and opsonization. This alternative pathway is so named because it was discovered after the classical pathway. Unlike the classical pathway, it does not involve antibodies. The alternative pathway is activated by contact between certain complement proteins and a pathogen. C3, constantly present in the blood, combines with complement proteins called factor B, factor D, and factor P on the microbe surface. The complement proteins are attracted to microbial cell surface material. Once the complement proteins combine and intact, C3 split into fragments C3A and C3B, as in the classical pathway. C3A participates in inflammation, and C3B functions in cytolysis and opsonization. It is common to draw more than one blood sample for laboratory tests. The blood is collected in tubes with caps of different colors. Whole blood may be needed to culture microbes or to type the blood. Serum may be needed to test for enzymes or other chemicals in the blood. Serum is the straw-colored liquid remaining after blood is allowed to clot. Blood plasma is a liquid remaining after formed elements are removed from unclotted blood. Complement activity is measured because complement deficiency may be associated with recurrent bacterial infections. The right figure shows how total complement activity is measured. Dilutions of the patient's serum are mixed with sheep red blood cells, RBCs, and antibodies against sheep red cells, NRBCs. Following incubation at 37 degrees for 20 minutes, the degree of hemolysis, bursting of red blood cell, is determined. Some bacteria evade destruction by complement by means of capsules, surface lipid, carbohydrate complexes, and enzymatic destruction of C5a. Some gram-negative bacteria, such as Salmonella, can lengthen the O polysaccharide of their LPS, which prevents MAC formation. Gram-positive cocoa release an enzyme that breaks down C5a, the fragment that serves as a chemotactic factor that attracts phagocytes. Because viruses hijack host cells to carry out viral multiplication, it is difficult for the immune system to inhibit viral infections without affecting body cells too. One way an infected host cell encounters viral infection is with a family of cytokines called interferons, IFNs. These are a class of proteins produced by certain animal cells. Interferons are host cell specific, but not virus specific. There are three main types of human interferons: alpha interferon, IFN alpha, beta interferon, IFN beta, and gamma interferon, IFN gamma. Some virus-infected host cells produce substances called alpha and beta interferons. Mode of action is to induce uninfected cells to produce antiviral proteins, AVPs, that inhibit viral replication. Gamma interferon is produced by lymphocytes, which causes neutrophils and macrophages to phagocytize bacteria, also involved in tumor immunology. Recombinant interferons have been produced, however, short-acting and many side effects. No effect on already infected cells. Here is antiviral action of alpha and beta interferons. First, viral RNA from an infecting virus enters the cell. Secondly, the virus induces the host cell to produce interferon mRNA, which is translated into alpha and beta interferons. Next, interferons make contact with uninfected neighboring host cells, where they bind either to the plasma membrane or to nuclear receptors. Interferons induce the cells to synthesize antiviral proteins, AVPs. AVPs degrade viral mRNA and inhibit protein synthesis, and thus interfere with viral replication. Thank you for your attention.